This is a completely new talk tonight because I wanted to talk about largely what we were working on at the HackSeek and progress, but it takes a lot of background, I think, to understand why it was so interesting to me. And so I started at Arizona State University 3 years ago. So a little bit, how many of you are students? Most people. Great, great. Right, we're all still learning, but some of us have to get graded on it. So you're done with classes for now. But please, I'm fine if you want to interrupt me during this, and I'll repeat the question for the recording. So I was a mathematician. I was trained in medical mathematics, and I got to do a summer research experience for undergraduates where we modeled tumor growth using systems of differential equations, and I just thought it was my first insight into bioinformatics, that you could use computing to solve really cool biological problems. And so I was, despite not being a biologist or a computer scientist, they let me into a bioinformatics program for my PhD, and I've never looked back since. So I largely focus on sex chromosomes in mammals, but really any sort of sex bias in gene expression and in any species that has separate sexes. We're doing some cancer work, but we're actually looking at recombination and cool stuff there too. So it filters through. All right, let's see. Great. Okay, so first I'm going to take you on a little tour of sex chromosomes, so we're all at the same place, and why they're so interesting and important. And I hope to convince you that understanding your system and understanding evolution is really crucial for being a bioinformatician, right? So one of the biggest challenges I have myself and I see with students is that we have all the data. We have all the data, and you can do everything with all the data, but why was it collected? What do you want to do with it? What's your question? What's the biological meaning? Why is it important? What are the biases that might exist? And for here, I really want to say that evolution is really important, and because people haven't been thinking about the evolution of the sex chromosomes, I think we've been doing genome alignment wrong. All right. So we'll talk about sex chromosomes, alignments, XY align, and next steps. So I didn't get to use my laptop, which has my HackSeek sticker on it. I can prove to you all. Right, and yeah, specifically a note about language. So I'm really going to try to be specific tonight and talk about individuals with an X and a Y chromosome, individuals with 2 X chromosomes. And for other work that I do, I'll talk about males and females, and that's typically individuals with an X and a Y chromosome. Males are individuals with an X and a Y chromosome who develop testes and make sperm, and typically females are individuals with 2 X chromosomes who develop ovaries and make eggs, and that's not gender. And there is a spectrum across sex, and there is a spectrum across gender, and while there is overlap, they are not identical. And so I just want to be really careful with that. So when I say male and female, I'm really talking specifically about genetic sex here. Cool. So yeah, I figured if we start at death, we can only go up, right? Okay, so we'll start with death. In the US, right, because it's the CDC. So leading causes of death for males and females. And so this is what's been reported. So anyone know the leading cause of death for males? Heart disease, right? Yeah, okay. Heart disease, almost a quarter of the proportion of people who die are going to die of heart disease. What about for women? The answer was men. Well, so since I'm trying to be specific here with males and females, I will say not exactly. But anybody else? Right, we all know symptoms of a heart attack, right? Like you see some guy clutching his chest, but in fact, heart disease is also the #1 killer of women. In fact, with different symptoms. So most of our medical studies have been done in populations of typical males, karyotypical males, and we know the symptoms really well, and we don't know the symptoms really well for XX individuals. We know that even the treatments are different. We know that aspirin responds differently in XX versus XY bodies. In fact, there's just a paper came out recently suggesting that, so you might have heard take a baby aspirin. Well, you might not have. Some people here might have heard that. I don't know at what age. I'm at an age. Anyway, take aspirin, right, to reduce your risk of stroke and heart attack. But it turns out it works pretty well to disaggregate the platelets in males, and it doesn't do so well in females to disaggregate the platelets. So there are some really interesting sex differences that we don't study. And right, we know heart disease is the #1 killer of males, but most people don't think of that as the #1 killer for females, right? We're focusing on death for like a few minutes here, and then we'll go up. Okay, right, cancer kills everybody, right? Proportionally a little more men than women, and most cancers actually tend to be sex biased towards men. So of the cancers that are actually still on the increase, melanoma is one of them, and it's increasing faster in males than females. So we think there might be some hormonal component to that. Number 3 is where we start to diverge. So number 3 killer of women is stroke, about 6% of women, and it doesn't show up until number 5 for men. And I'm sorry, I'm merging males and females and men and women here because we're working with humans, and typically these studies don't actually disaggregate them very much. Have you heard of broken heart disease? Star Wars. Have you heard of Star Wars? What does that have to do with broken heart disease? Carrie Fisher, Princess Leia, passed away recently, and her mother died just 2 days later, had a stroke. It's actually called broken heart disease. And we know that it occurs more often in women than men. We don't exactly know why. Number 3 is unintentional injuries for men. Turns out that doesn't show up to number 6 for women. I unintentionally injure myself all the time, but luckily haven't killed myself yet. Number 4 is very similar, chronic lower respiratory diseases. Number 5 is also a really a different one. So Alzheimer's is the 5th leading killer of women. And we used to think, right, okay, so where does it show up for men? It's not even until number 8, right? And it's less than half the proportion as for women. So we used to think, well, if it's an age-related disease, and typically women live longer than men, then they'll get more Alzheimer's because it's an age-related disease. But it turns out when we do age-matched controls and we account for all of that variance, women still get Alzheimer's at higher rates than men do. We think now there's an immune system component. And for me, that's really interesting. So we know autoimmune diseases occur more often in women than men. We also know that the X chromosome is enriched for these cancer testis antigens and immune system genes. So, right, we think maybe there's some difference in copy number of X chromosomes. It should play an important role in this disease. So, of course, with all of this sex bias in disease, people are looking at the genetics that are sex biased. Oh, right? The X chromosome is reported, not even analyzed, reported in about 33% of so-called genome-wide association studies, right? It's so underreported, there's people now working on XWAS, like including the X chromosome, just being like, hey, look at this 5% of the genome that you're just throwing out. That's actually, in fact, extremely sex biased. The Y chromosome is almost never included. And methods, so all these bioinformatic methods that we're working on are really meant to work on diploid chromosomes that are typically diploid in individuals. And that doesn't work so well with the X chromosome, right? So let me talk to you a little bit about the sex chromosomes now. Okay, yeah, we're done with death. Okay, so we can get back. Okay, so here's a karyotype. So right, it's a picture of bands, a human karyotype. This GIMS is staining, and you typically get one copy from each chromosome from each genetic parent, and they all are homologous. They can recombine across their entire length, except for the sex chromosomes. So typically, genetic females will have 2 X's, and typically genetic males will have an X and a Y. So that's our starting point. And I'm going to come back to this slide a couple times. So the X and Y chromosome, we have really good evidence from comparative genomics that they used to be completely homologous, nearly identical, indistinguishable from each other. There used to be a pair of autosomes. They could recombine with each other over their entire lengths. And over about 160 million years, so in not all mammals, just in therian mammals, so those are the marsupials and us eutherian mammals. Sometimes we're called placentals, but that's a misnomer because marsupials make a proto-placenta. We'll call it placentals, yeah, right? Placentals and marsupials, the pouched ones. We share the same, largely the same X and Y system. And our genes on our X chromosome are autosomal and synthetic. Gene order, gene content is conserved on autosomes in other species. So we have really good evidence that they were autosomes and then over 160 million years became really differentiated from each other. So the human X chromosome now has about 155 million sites on it and give or take 1,100 genes. How do you define a gene, protein coding unit, blah, blah, blah? 1,100, okay. The Y chromosome, about 60 million sites, 27 unique genes. Some of those have many, many copies. Some of those might have 76 copies. Some of them, many of them have become testis-specific, but there's some that are still shared between X and Y. Right? And so just keeping in mind this shared ancestry is really going to be crucial later. So there's one other part, just to keep you up to speed, is that so marsupials and us, eutherians, we share these ancestral sex chromosomes. Where's my mouse? Oh, there we go. But after marsupials and eutherians split from each other, all eutherians, so dogs, elephants, dolphins, whales, us, cats, dogs, mice, we have a large translocation to our sex chromosomes. So 30% of our sex chromosomes was added more recently, and it's autosomal in marsupials. So here's another region. So these regions started to differentiate from each other, and then we added these whole identical regions to our X and Y. So there's at least 2 times where our X and Y were nearly identical to each other. And just the really cool thing is, so some comparative genomics work I did at the end of my PhD was showing that when we do comparative, we say which genes are synthetic, which genes are on the X or the ancestral autosome. We can show the Y chromosome has lost about 90% of the gene content it used to share with the X. So it's lost a tremendous amount. It's gained a few genes, but not very many, right? There's 27 unique ones there. So how did they become so different? This is all leading up to important stuff. And I guess what you all appreciate that doesn't always come across is I am a bioinformatician, right? I get to show these pictures, but literally everything I do is on a computer. So right, my university came to me and said, like, can you send us a picture of you working in the lab? And I was like, yeah, I mean, yes. It's me on the computer. And they were like, yeah, but like doing something. Yeah, I will open my terminal. It's exciting to us. But I think why bioinformatics is so great is when we get to figure out new things about biology. Things other people couldn't do because they had all this data, but they weren't asking the right questions. They didn't know exact ways to put it. Or what do you do with a big pile of data, right? There's this, what was it, XKCD cartoon that came out recently? It might have been someone else, right? And they're like, I have little data, and I have big data. What do you do? And you give it to a bioinformatician, and they come back with like 1/10 of the data. And they're like, here you go. I found out something new for you, right? But we're asking and answering biologically meaningful questions. Okay. So here's our ancestral sex chromosomes, and we're going to put them, here's our little cartoon. And A can recombine with A, and B, and C, and D, and D, and E, and E, right? So they can recombine across their whole length. Recombination is a pretty good thing to have. Homologous recombination can fix errors, insertions, deletions, mutations. Great thing to have. Okay. But at some point, we hypothesized that there was a sex-determining region. So it was called testis-determining factor, sex-determining region. Scientists thought SRY1. And individuals who inherit that allele will go on to develop testes, and individuals who don't inherit that allele will go on to develop ovaries. And it's not sex chromosomes yet. It's just an allele. And we see some systems that just have allelic sex determination, right? So you don't have sex chromosomes until you have a second step, which is having a sexually antagonistic allele. So something that's beneficial for males and harmful for females in this case. So that means you would, if you could want, if evolution could want, but it doesn't, because it's cruel and unfeeling and it doesn't want anything. But individuals in which SRY, so being male, is always linked to this beneficial thing for males, will have higher reproductive success than individuals who have them separated from each other. It'll sweep through the population. One way that can happen is an inversion, so that if you make testes, you always have the thing that's good for testes producers, and if you make ovaries, you never have the thing that's bad for ovary producers. So if there's strong enough selection there, it'll sweep through the population. You have an inversion. Now you have sex chromosomes. Yay, look at that. They're so pretty. Okay. But now this thing, recombination, that is so fundamentally important for life. Why are we diploid, right? Recombination. We have now suppressed it between X and Y, which is a little ironic because we call recombination sex, and now we have sex chromosomes that can't recombine anymore. But there was more than one inversion. Okay, that's my joke. Nobody liked it. Okay, yeah. Okay, there we go. So we have one inversion. We have at least two inversions. And what this means is that now we can bioinformatically, computationally, determine when those inversions occurred. Because the regions that stopped recombining early are going to start to accumulate mutations between the two pairs. We're going to call, they're homologous pairs now. We can call them gametologs because they're not paralogs. It's not a duplication, but they are separate genes now. They're accumulating mutations separately. And these ones have been accumulating differences from each other for less long. So we can actually count substitutions between the X and the Y pair. And so that was done. So Bruce Lawn and David Page published this in 1999. And along here is the X chromosome from the short arm. This is the regions that were added recently. And these are the older regions. And they, looking at synonymous substitutions, so counting substitutions that we don't think change the amino acid between X and Y, found some genes that had a tremendous number of differences between X and Y, some that had a lot of differences between X and Y, some with some differences between X and Y, and some with very few differences between X and Y. So they hypothesized that there were 4 what they're going to call evolutionary strata, because we're discovering new things, and so we build on language that exists before. So it's like geologic strata in time, going down, being older, only we'll swap it, right? So it's like this, where these are the youngest to the oldest, and here we have the youngest to the oldest strata, potentially inversions. And then again, what we can do is when we have the sequence, up on the top is the sequence and gene order of the human X chromosome, and on the bottom is the sequence and gene order and orientation of the human Y chromosome. And you can compare those and actually identify inversions and infer parsimoniously, so with the least possible numbers of inversions, how many occurred, and you can actually identify how many might have occurred between X and Y. So for the most recent, they identified a 5th evolutionary strata using this method. When I was a postdoc, I was working with some computational scientists at UNT, and we developed a new method to look at sequence similarity. And we proposed that there are at least 9 evolutionary strata. We can go into those methods later because I want to get to the new stuff we've been doing. But that means there's also, okay, so we have these evolutionary strata. That means X and Y have differentiated. They can't recombine in those regions anymore. But at least for us, we need chromosomes to pair and recombine so they can segregate properly. So we can have in sperm 1X or 1Y. So there's pseudo-autosomal regions, PAR. Here's the tip. That's the leftover of that big translocation, pseudo-autosomal region 1. And there's a second one that's human-specific. Chimpanzees don't have this one. It was duplicated just in humans from the X to the Y, pseudo-autosomal region 2. So there's these 2 pseudo-autosomal regions that can still pair so that you can have proper segregation. those regions are indistinguishable between X and Y. Okay. And like we showed with the, there is this inversion that actually separates, it occurs right in the middle of a gene, which is pretty neat to me, that it broke this gene. So it's a gene called XG, it's a blood group antigen. And the X chromosome is a fully functional copy, the Y chromosome, half the exons are several megabases away. Oh, and the cool thing, just side note, is in one of those inversions, it brought SRY immediately proximal to our pseudoautosomal boundary. SRY doesn't live right there on the Y chromosome in other species, but it does in us with some interesting medical implications. Another thing to keep in mind is that, so we talk about typical. I will always talk about typical chromosome numbers, typical karyotypes, because which one of us is normal? I'm not normal. None of you are normal, right? We have typical things about us, but especially on the sex chromosomes, other than chromosome 21, X and Y have some of the highest atypical karyotype numbers in the genome. So Turner syndrome, which is just a single X chromosome, is about 1 in 2,500 live female births. Klinefelter, which is 2 X's and a Y, and developing typical male gonads. There's also XXY that develops ovaries, but it's not called Klinefelters. Biology is messy, right? Klinefelter syndrome is about 1 in 1,000. So I give these numbers and I think about ASU with 70,000 students. And we have several students that have Turner, have Klinefelter. If we think about our genomic studies, right? So when the 1,000 genomes started, you had 1,000 people, maybe 50-50 male, female. Maybe you would have an individual with Turner or Klinefelter. But now we're getting larger and larger data sets, 100,000, right? The genome 10K, 10,000. 200,000. We want more data. We always want more data, but when we get more data, then we have to understand all the biological variation that exists and how that might play out when we're actually doing our alignments, when we're doing our variant calling, when we're doing our sequencing experiments, RNA-seq, right? And so alignments, right? What on earth does sex chromosome evolution have to do with genome alignments? And I'm actually really hoping to argue that evolution really matters for this, largely because of misalignment of reads. So here's that karyotype again, right? We're going to come back to it. So you get 1 copy of each of those chromosomes. Typically, you'll have 2 copies of each of them. Maybe you have like a small duplication, copy number variations, blah, blah, blah. Typically, you'll have 2 copies. But when we're doing genome alignments, do we align to 2 copies? No, we don't, right? We just align to 1 copy. This leads to a big problem, which is reference mapping bias, which a lot of people are working on, right? Because if you're allele, you're doing short read sequencing, and you have a heterozygous site, and your reference is 1 allele, but your individual is heterozygous or has a site that doesn't match the reference, you're not going to map them as well, because what do? We make sequence similarity algorithms that will try to match them identically. So there's been a lot of effort looking at reference mapping bias because we do this, because we do 1 copy. But we include both X and Y in our reference human genomes. We include both of the sex chromosomes, even though we only include 1 copy of every other chromosome. So what does that mean? Well, we have some expectations for relative depth. I showed 8 here because I didn't want to make up the chromosome. That's actually chromosome 8, and that's actually X, and that's actually X and Y, right? So if we think about depth, you've done your sequencing experiment, and you think about mean depth across the chromosome. So you've taken the genome, chopped it up into small pieces, hopefully not done too much amplification that's going to mess up stuff, and then you sequence it, and you try to align it back to the genome, and you have average depth across a chromosome. So in an XX individual, you'll expect the autosome to X ratio to be about 1. You should have 2 copies, typically, of any autosome and 2 copies of the X. So that autosome X ratio should be 1. Trivial. In an XY individual, it should be 2, because you'll have twice as many copies of the autosomes relative to the X in a typical XY. It seems, sometimes when I think about this, I feel like I'm being way too simplistic, right? But let's think about AY, and I put 0 there even though it's actually infinity, but we can, I'll show you why, because the plotting didn't look good when we did it the other way. It was really hard to interpret, and so it's easier to interpret this way. Which will tell you then, spoiler, the y is not 0. Okay, and then the AY ratio should also be 2. Mean depth across the y relative to any autosome that you choose. But is it, right? Well, first of all, we know it absolutely isn't going to be. So here's a dot plot. So this is a plot from the genome paper that is the DNA sequence of the human X, right? They announced the sequence of the human genome. The human genome was published a couple years before we had the X chromosome. I mean, it's not like it's that important. Okay. This is a pseudo-autosomal region right here. It's just one-to-one dot plot between here's the Y and here's the X. And so we have the pseudo-autosomal region almost one-to-one. This is that X added region, well, at least part of it, that still has a lot of sequence similarity between X and Y. And this is something called an X transposed region. Another cool thing that's human-specific, duplicated from the X to the Y. And it's really highly syntenic. So we at least know these regions are really highly, there's some, you can see these tiny, it's hard to see the resolution, but there's little genes here and there that are similar and aligned between X and Y. So there's known sequence homology that we don't take into account at all. Because what we do is we have a reference that has one copy of every autosome in it and has a copy of the X and a copy of the Y. So what happens if you take an XX individual and align them? Here's my cartoon, yeah, reads. So we take chromosome 8, for example, and we squash them all together and we align them to chromosome 8. And I should have dropped some of them out, right? There's some reference mapping bias. We've lost a couple of them, but largely we're aligning most of it to chromosome 8. It's not going to happen for the X chromosome. Because of shared evolutionary history, you're going to get some of the reads from the X mapping over to the Y chromosome. More in the pseudo-autosomal regions. I didn't make the Y bigger, but pretend it's scaling there. Okay. So we're going to have more in these tips and some in the X transposed region and some in those regions that are still shared, the genes that are conserved and shared between X and Y. So even though we know this person doesn't have a Y chromosome, we have reduced coverage, we have reduced depth, and reduced quality on the X chromosome because our standard is to align to a reference that has X and Y in it. Okay? So then what are the problems if you have an XY individual? Well, you know, your autosome is still going to be fine, and you're going to map between X and Y, but you have half depth, first of all. And then especially for the pseudo-autosomal regions between X and Y, they're nearly identical, so they're going to map in between X and Y because they're literally recombining regions. It's exactly the same thing here, only if I split up the reads if I had 2 copies of chromosome 8. But we don't analyze them that way. We don't take out the pseudo-autosomal regions and analyze them differently. So what do we think about relative depth? So these are actually plots that the program will make, but we're going to show these and then go back. So what do we think? So depth around 2 for 19 versus X, or should hopefully not have almost any in females. So for males, this is high coverage whole genome sequencing. We're not quite at 2 for our, the reason I switched to chromosome 19 here is that it has one of the lowest aneuploidy rates of any chromosome in the genome. That's also fairly small. So it's fast. So my bioinformatics hat was like choose something fast, and then my biological hat was like choose something that doesn't have a high rate of ploidy typically in the genome. So that's 19. So we're not quite at 2. Or even at 1, right? We're here in some weird zone, but if it differentiates between males and females, then we're okay. And it turns out, so our males are going to cluster here, and our females are going to cluster over here, with many of them having a non-zero number of reads on the Y chromosome. To me, if you're XX and anything's mapping to the Y chromosome, that's just bad analysis, because you don't have a Y chromosome in those people. So all of those reads are messing up any sort of downstream analysis that you're doing if you're thinking about an X chromosome. So we've done some different experiments with exome capture, low coverage whole genome, where you have really some regions of the genome that aren't going to be covered at all and some that are covered higher, and then high coverage whole genome, where at least you hope most of the genome is covered. And in fact, your expectations are really different based on your data type, based on whether you're looking at exome, and we haven't dug yet. So this is really new stuff, right? We haven't published yet. We're hoping to soon. But our exome capture data looks very different than the low or high coverage whole genome data. So it could have to do with biases in how the capture kits were developed and what they're pulling out that we'll hopefully get to. Right? Okay. So what can we do about it, right? Should we, well, one, I hope we should care about it, because if we're doing any sort of medical genetics, if we're doing evolutionary analysis, if we're looking at selection, diversity, population history, I want my variant calls to be true, and I want my gene expression to be true as much as possible. And so we want our alignments to be good. And so at HackSeq, right, I had this idea. How many people went to HackSeq last year? I mean, yeah. And you all let me come, which was great. So I submitted this idea because I've been thinking about it for a while, and we had this great team. So Bruno's over there, and really been since then taken on by Tim Webster, who's a postdoc in the lab who's working on it. So over 3 days, we set up the problem, we thought about the problem, we tried to solve the problem. We realized 3 days was not enough. It's been, 6 months, but that's fine because we want to make it good and we want people to use it. So what it will do is actually compute this relative depth on your input files. Oh, I will hurry. I'm sorry. Okay. And then it will say, so instead of using a standard reference genome, if you have no Y chromosome, mask the Y chromosome. So if you have evidence that there's no Y chromosome in your set. So we're going to infer it from your input BAM file. We're going to infer that you don't have a Y chromosome, mask the Y, strip the reads from the Y, and realign them. If you do have a Y chromosome, filter out the pseudoautosomal regions, strip those reads and realign them, because at least we have higher variant coverage or higher confidence in calling those regions. Okay, so then what it should do is go from our standard reference in an XX individual and say, look, there's reads there, they shouldn't be there, strip them and map them back where they should be. And in an XY individual, it'll strip the pseudoautosomal regions and map them back. So at least we'll have higher coverage in these pseudoautosomal regions. And so what does it actually do? So let's look at an XX individual. So I want to show you how it actually looks on real data. So if you haven't looked at your data, I hope that you will go back and look at it. And I hope you're looking at the X chromosome, at least. I'm going to flash forward and back. I tried to, okay, I didn't try. I thought about making a GIF, but I didn't. So anyway, we have this. So there's a pseudoautosomal region. This is read depth in Windows. And you can see in an XX individual, this individual doesn't have a Y chromosome as far as we can tell from the sequencing data. And we're doing a really poor job of read depth in the pseudoautosomal region because it's split in half. And we're also doing a less good job in the X transposed. And then there's the second pseudoautosomal region is eensy-weensy. It's 328 kb. It's right there. Okay, it's tiny. And then if we apply XY align and we reassign the reads or realign the reads, then we fix a lot of that problem. So we go back. Look. Okay, so we look at pseudoautosomal region. Oh, look at it goes up. Yeah. Okay, and then we look at the X transposed region, and it was down and it's up and it's down and it's up. Haha. And then and the PAR2 even, this tiny 328 kb, right? It's tiny in the whole scheme of the genome, but there it's down, and then we fix it. We're also fixing other things, but we're not tracking all the reads here, and our eyes aren't really good at seeing them. But we also can look at, this is map quality, right? So previously, we just did really, really poor with quality in those regions where you're aligning things that shouldn't be on the Y chromosome to the Y chromosome. You don't have any confidence in calling variants in these regions. So we're going to do before, after, before, after. So certainly in those regions, we have gone back to the quality that we should have simply by using the right reference genome. Isn't even, on the scale of single nucleotide reference mapping bias. We've just been messing it up by having a whole extra chromosome there that we shouldn't have. Here's read balance, or reads that are coming from the Y chromosome in an XX individual, as far as we can tell. So all of those are faulty. None of those should be there at all. So what's this mean for XY individuals? We have less power to fix some of the things that we know are wrong, but at least we know they're wrong. And one of the things we can fix is variant calling. So one of the things XY align will do, will go across, compute map quality, compute read depth, and give you bed files out with the regions that you should be looking at. And so prior to doing this, this is read balance from 1 to 0. You shouldn't be anywhere near 50% because that means you're diploid. If you're on a Y chromosome, you shouldn't be diploid because you should only have one copy of it. There are XYY individuals, also XXYY, less typical. But most of all of this is just poor mapping due to low quality regions. And then after running XY align, our read balance goes to 1. And I will finish up real quick. So we've tried applying this to RNA-seq also. So Kimberly Only and Sarah Brotman, a graduate student and an undergraduate student. We were talking about microarrays earlier. This aligning between X and Y being a problem is a problem for microarrays. When I was a graduate student, the probes that they had for X and Y were not always specific to X and Y because they were built off of X, built off of Y, and we had to go through and identify just the probes that were X and Y specific. And so I thought when we got to RNA-seq we would do better, but we're not because we're including X and Y. So we can do gene expression before, gene expression after, we do XY align, and we identify several genes as being differentially expressed when we fix your reference mapping bias issues. And in fact, all of these genes that are Y-linked previously were not differentially expressed between our XX and XY samples. So previously, the RNA-seq analysis was telling us there was gene expression for all of these Y-linked genes in XX individuals. Oops. Right. Okay, so next steps, it'll do a lot of cool stuff, giving you depth and map quality, genome masks that are not just for variants, but actually a whole region that you feel that perhaps you shouldn't trust, shifts in heterozygosity. It's up on GitHub, right? Of course, open science, everything we do should be made publicly available to everybody. It's got a cool logo. I did not design, Whitney designed it, right? So take-homes are that, right, evolution really matters. And if we're not thinking about it, then we're just going to use all the chromosomes, throw them in there, and mess up our read depth. And hopefully we have this tool now. It's available now. We haven't formally published it yet, but in open science, that's kind of trivial. We're doing some other projects, notably like cool Gila monster work, different cancers. But really, thanks to the people who've been working on this project, my PhD mentor, for the HackSeek, for letting us really sit and think about this for 3 days. I don't get 3 days to do anything. It's really cool to have those 3 days to just think about one thing. And thank you all for being here. Thanks. Yes? So the question is, have we tried de novo assembling X and Y and also for the autosomes? And I agree the next stage of genomics is really going to be doing de novo assembly, but with the short reads, it's impossible for the Y chromosome because most of the Y chromosome is heterochromatic or high copy number variation. And with reads of 150 base pairs, you can't do it. Even reads of 10,000 base pairs, you won't be able to de novo assemble it without getting the PacBio, without using super long reads. But I do think, right, we assume everybody has, all of their chromosomes intact, but chromosomes break, chromosomes rearrange. I do think de novo assembly is where we're moving, and that might help, but it's always going to be harder on X and Y because you have to be able to say, did the reads come from X or Y, even when you're de novo assembling them. So for people who are doing Y-linked stuff right now, they'll flow sort get out the Y chromosome and then sequence just that, or at least they'll amplify the Y chromosome by itself because you can size sort it. So at least in species with X and Y that are really different sizes, you can flow sort them that way. But in species with X and Y that are pretty similar, I don't know how we'll distinguish that so well. Until we do, right, I have all of the money in the world and I can do all the sequencing. Yeah.
So I was thinking, so there's a lot of other reasons the genome has problems with repetitive sequences, like all the parallel, for example, in MS2 that always have homologs that you see the same thing with like approach help with some of the pseudogene issues.
So the question is, could this help with pseudogene issues or copy number variants? I think in a way, yes, because if you're just doing your broad genome for any individual, you can identify in that individual regions that are higher or lower mean depth than the rest of the chromosome, because you'll get bed files out. You can do those plots, so you could either manually inspect them or write a Python script to do it. And also quality, you can get the map quality scores. So for things that are polymorphic in the population, we actually really do think this will be helpful for more than just X and Y. So the assemblies of the X and Y chromosome are pretty good. So there's a whole group, a consortium that sequenced and assembled the human X chromosome, and a whole separate consortium. It took almost, this may be overselling a little, but almost as long to sequence and assemble the human Y chromosome as it did most of the rest of the genome, because it's so highly repetitive, heterochromatic, and really difficult. And it's pretty good, but we still, how much variation is there across populations? There have been some forays into that, but I think that's going to be a new avenue for research with the new technologies that are coming out. I haven't looked at 10x chromium data yet. We have, we're waiting on some PacBio data, but not PacBio. Sorry, 10x, the short read supernova, so not the chromium, but the supernova assembly for the Gila monster. And so we're going to be looking at that, but that's challenging because we don't have a reference at all. We're just trying to make something. So I haven't yet got that data, but would be happy to work with anyone who does. All right. Thank you.